it up again. Awesome, I love it. All right, well, welcome to the FRS Spring Seminar Series. So I will provide a brief introduction, but if Ben wants to provide more, I'll let him provide more of an introduction. So today we have Dr. Ben Blonder from UC Berkeley speaking with us about some of the work he's been doing on Aspen. I grilled Ben this morning on kind of his background, not to actually provide the introduction just because I was interested, but then I was like, oh wait, I can actually use some of this information in the introduction. So very briefly, Ben grew up in New Jersey and then went to Swarthmore College and did physics, bachelor in physics. And I asked him, were you always interested in biology, or when did you get interested in biology? And so kind of, he said he was interested in complexity. And after undergrad, then he was able to spend some time outside and realized there's lots of complexity in the tree species and plant communities, and got more interested in ecology and that aspect of science. And then after working for a few years in some different jobs, went to University of Arizona where he got his PhD and worked with Reinecker there. And I think that body of work is where I first got to know Ben just through reading. I'd never met Ben until this morning, but I was really interested in his ability to combine kind of theory and quantitative methods to understanding the complexity of leaf anatomy, leaf traits, and in particular leaf hydraulics and veination. It's one of my interests, and that's why I got interested in some of Ben's work. So this seminar series, Sarah and I both had some interest in having Ben here, and so we co-hosted Ben. And so I've had a really great time getting to chat with Ben a little bit, and glad I actually know you in person now. And so I hope you all enjoy the seminar. After the seminar, we will do a social at Avos. So if you'd like to meet Ben and talk a little bit more, you can join us at Avos. We'll probably show up there around 5:30. So feel free to join us. Otherwise, I will turn it over to Ben and let you take over.
Well, thank you, Troy, for a really kind introduction. I'm going to be talking today about none of the things he mentioned, but actually a tree species which I fell in love with probably the first year I ever spent any time in the Rockies, which is Quaking Aspen, the tree species that you see illustrated up here on the slide. This is a picture from western Colorado in the late spring. And for those of you who don't know it super well, it's one of the more widely, in fact, perhaps the most widely distributed tree in America. These are polygons where non-naturalist people have reported seeing the species, and you can see up into Alaska and even beyond where I cut off the map here. And down into central Mexico, we have the species out on the west coast, all the way out on the east coast, and we have all these other, small parts as the tree line disappears up in Nevada, and then down through the Appalachians as well. So it's a really widely distributed tree species that touches a lot of people's lives. And I think it's a really interesting test case for asking some questions about the futures of trees under climate change and land use change scenarios. To give you just a little bit more background, this is a picture that does have some aspen in it. Does anyone recognize this photo where this location is? What take it is? So this is in California. This is in the Sierra Nevada. This is Donner Summit, where the Transcontinental Railroad goes over, and these are actually some of the snow sheds and the tunnels produced by the Chinese laborers that built this system. And then this is Donner Lake down there. And there are aspens here, although it's hard to see it in the black and white photo from a long time ago. This is a more modern view of the same location with the train tracks full, but you can see a little aspen stand.
Hey Ben, I'm so sorry to interrupt. I was just wondering if people in the back could hear very well, or if we should get them mic'd up with the room system.
Thumbs up. A little louder. I'll do my best to be a little bit louder. I'm not naturally a loud person, so
I can get you hooked up. Yeah.
Sure, I'll keep going in the meantime though, just for the timing. So just a bit of background here. So yeah, you can see aspen here in this location. It's a small component of the forest, but an important one. I also show this photo because for what I said about aspen forests touching a lot of people, Donner Lake, it turns out, the name for Donner Lake actually has a Washoe name as well, the indigenous people that live in this area. And their name for this lake is actually basically Aspen Lake. So it's a species that really connects to a lot of different people, and we see this throughout the range as well. One of the reasons that I think it's worth studying this species is that in many locations we do see dieback of aspen forests. We do see places where we're losing these forests. Anyone recognize this picture? It's another trial of Grand Mesa. Luke is correct in this case. This is the Grand Mesa. Bear with me for a second. Doesn't look like that's working.
You keep going.
All right. So you can see here in this picture, there are these big white areas, and these are locations where we see canopy dieback of the species. So this is happening in many other locations as well, and is of course a pretty big concern from a management and conservation perspective. We think there's good reason to believe that these are historic trends as well. So here you can see a picture from 1872 as part of the PAL survey of the West. This is in Utah, Boulder Mountain. These are all aspen trees here in the foreground. And if you go to repeat photography, this is the same location in the late 1990s. And you can see there are some mature aspen stands, but there's a lot more conifer competition and perhaps the decline of these aspen stands. You can see a lot of adult tree mortality in these locations as well. So it's a big landscape change. And if you go back to historic records of land cover to the extent that we're able to make them, even here from the 60s up until the 1980s, you can see aspen cover in Utah has decreased very substantially even over that time period. So it's not just a present day sort of decline, but really a longer term century to multi-century type of process. And if you look forward to the future as well, you can build species distribution models based on environmental variables, predicting the future of the species. And this is one done by the Forest Service in 2015. And the red areas are places where it's bad news for aspen. And Western Colorado here again, you can see there's a lot of areas where we think there will probably be further losses of the species. So that's all known, and just some background for the talk today. What I want to ask is this question about whether or not all aspen populations are really equivalent to each other. If all individuals necessarily respond in the same way to a given stressor. So here's another Colorado picture, and these are all aspen trees here until you get to the conifer land up here. And phenology-wise, right, they're not all changing color at the same time, suggesting that at least this phenotype differs across the landscape. And then because of these sharp boundaries, one might imagine that genetically these trees are also differing. So we'll explore that in this talk. It matters for a lot of reasons because many things we do in forests sort of assume that a individual will respond in a certain way. So for example, if you're coppicing or thinning a forest and you have some aspen coming back, you might be selecting for certain genotypes relative to others. If you have a fire coming through, similarly, you get sexual and asexual reproduction afterwards. Who is that? Are they the same things that you used to have? Is it genotypes from somewhere else? Are they adapted to the new conditions? Are they not? If you put up a fence and you get the herbivores out, are the trees that are going to come back in an herbivore-free location going to differ genetically from the ones that you used to have? Who knows? And then lastly, as we talked about before, if you have drought or big climate stressors, who's going to be able to survive that? Who are we losing and who are we gaining? And for novel diseases as well, you can see some fungal disease here and then some scale insect disease here. There may be resistance that may spread through a population. There may be selective sweeps. We just don't know. What is the case, though, is that if you look across the species range, there is very substantial variation in phenotypes of aspen, suggestive of a lot of genetic variation. So just to illustrate a few photos for you here, here's some aspen in Alaska. Here's some out in Western Colorado. This is a little Krumholtz population here. This is probably 3 individuals on the top of a mountain. That's it. Here's in North Carolina, looks a little bit different. Here's in Texas in the Davis Mountains near the border, pretty scraggly, growing on a talus field. Here's in Mexico, where aspen is actually habitat to thick-billed parrots. And then here is down in Jalisco, where the tree gets much darker colored bark. So very different trees in these different locations. And then at smaller scales, we also see this sort of variation. So just to illustrate this on a gradient across California, here are 3 sites that we recently visited. This is Lassen up in the north. You can see some trees here. Here's Sequoia in the central Sierra Nevada. And then down in the very southern part of California in San Gorgonio, some very different looking trees. So, we have a ton of phenotypic variation. We also know that there's a lot of genetic features that should impact aspen phenotypes. And I'll highlight a couple of the interesting genetic features about this species next. One of the first is cytotype or ploidy level variation. So this basically refers to the number of copies of a chromosome that an individual has. And as the ploidy level increases, the phenotype can differ due to gene expression differences and screening of certain phenotypes, regulatory differences, etc. In Aspen, we can get diploids and we can get triploids. And typically the triploids have reduced fertility because of the difficulty that they experience when they segregate their chromosomes. And just to show you what this looks like, just for a visual illustration, so haploid, 1 copy of each chromosome, diploid like us, 2, and so on and so forth up. And if you do a chromosome squash in Aspen, you can see here a difference here where we have a diploid on the left, and on the right, you can see sort of a larger number of chromosomes in the triploid when we catch them during meiosis. So that's physically what's going on here. We know as well from previous work that triploids are common throughout the species range, but they're more common in some places than others. And in particular, these little pie charts, the fraction of red is the fraction of triploids. Work from Karen Mock has showed that in the Southwest, we seem to get more triploids than diploids. And then more recent work by Roscoe Essen and others has shown a pretty similar pattern, although potentially some more complicated things up here in the Great Plains of Canada. So that's one thing about aspens that's different than many trees. A second is that there's sex variation. So aspens are dioecious, meaning that they can have male or female individuals, and on a given individual, you typically don't get both. So these are different stems from different individuals, and you can see them during flowering with either the presence of pollen-producing structures or not in these photos. It turns out, though, that the control of sex in this species is also not super strong, and it's actually possible as well that you can see flowers that have a mix of both male and female characteristics. This is a very old paper where they call them bisexual flowers, but you can see here that the fraction ranges from about 4 to 20%, depending on the population being studied. So sex is sort of a continuum here rather than a binary in the species. And there's also variation in clonality. So we're probably familiar with the idea that aspen grow in big clones rather than just as single stems. They do sometimes, but not all of the time, and it depends on where you are in the world. But certainly in places like Utah, you can get very big clones like this one, Panda, which we think is the largest tree individual in the world. In our own work, we've done some genetic analyses in Western Colorado, where we've looked to map out the size and distribution of clones. And here, what I'd like you to do is look at these different grids in different watersheds, and the background colors here indicate genotypes that are the same. So if you see the same color, that indicates one genetic individual. And what you can see is that there are some spatially extensive clones like the ones you see here, but there's a lot of interdigitation as well, and sometimes much smaller clones too. So I should mention here spatial scale here. This is 60 meters from here to here. So this is sort of, like football field size-ish, maybe a couple of football fields in size, but there's a substantial amount of variation in what the actual genetic makeup of a forest is when we look over fine scales. So that's some background, just things that would be relevant for understanding the future of this species. One of the first things we were interested in was, do any of these features influence the population dynamic outcomes for aspen trees? So to get at this, we set up a plot network in Western Colorado near Crested Butte, and we actually put about 500 plots in on the landscape distributed across a couple of different watersheds. And we did all the genetics on each of them. And then we revisited them in 3 different years, 2018, 2020, and 2023. And we did the full sort of demographic workup, growth, mortality, reproduction, suckering, et cetera. And what we then did after that is we built integral projection. So this is a demographic modeling approach where you can estimate the long-term population growth rate trend based on all of the different vital rates that go in underneath the hood. They're typically very difficult to fit because you need tree-level data. But in this case, we do have tree-level data for about 10,000 individuals, which was a lot of work in the field, but we did it. And even though you do a lot of work, the final thing you get is just, well, one of the things you get is this number called lambda, which refers to the population growth rate. If you get a value of 1, the population is stable. Less than 1, the population is declining, and greater than 1, the population is increasing. There's a lot of math that went in. I'm not going to talk about it, but if you're curious about the details of the models, we could talk about that afterwards. What I would like to show you, though, is the consequences of this modeling exercise. So we're going to be looking at some scatter plots of lambda versus sex, and then versus ploidy level as colors. in areas that have low recruitment or high recruitment, and then on north-facing slopes and south-facing slopes. So sort of our two big genetic features, ploidy level and sex, as a function of basically browsing pressure or land management, and then topographic position. What we see is the following. We see that on, sorry, I have this backwards. This is the first time I've given this talk. This should be south up here, north down here. So on north-facing slopes, the populations are pretty stable. No matter what your genetics are, everybody's got a lambda of about 1, so the populations are just hanging out. But as you go towards south-facing slopes, you begin to see a lot more variation. In particular, you see a lot of values of lambda that are below 1, indicating on the south-facing slopes, we predict that these populations are substantially contracting. We also see that in high recruitment conditions, there's a slight but not super strong effect where these populations are doing better, which is fairly unsurprising. More recruitment, more success. We also see that the diploids, the blue points, tend to be above the red points, suggesting that it's better to be a diploid than it is to be a triploid from a population perspective. And we also see that if we compare the females to the males, the left versus the right, there's not much of a difference on the north-facing slopes. But on the south-facing slopes, it looks like being a male, there's a lot more variation in what could happen to you. If you're a female, it's definitely bad. If you're a male, it might be bad or it might not. So to summarize all of that from the outcomes of these models, triploids do worse than diploids, especially on south-facing slopes. And females tend to do worse than males under low recruitment. And in general, it's just really bad to be on a south-facing slope, which I think is probably unsurprising to most of us who have been out on the landscape. So that lets us get some estimates of the population trends at these particular locations. But we might also wonder, well, what about the entire landscape? Could we map out these lambda values across the landscape? To do that, we would need to have all of these different features measured across the landscape. And things like topography, that's quite easy to do. But ploidy level and sex, you would have to do a lot of DNA sequencing work. So what we've proposed in recent work instead is maybe we could remotely sense those things instead of actually having to measure them from sequencing. So to do this, we flew an aircraft over this study site, the Neon Airborne Observation Platform, which has a hyperspectral imager on it. And it's quite a lot of work and money, but somebody will pay for it. And we can then train a machine learning model to predict those genetic features based on the spectral data. Just to illustrate the sort of spectral variation you see across the landscape, these are 5-meter pixels in this image here. This is a false color representation. You can see here in the Aspen classified pixels, there's just a lot of false color variation. So there's a lot of stuff going on in the forest from a spectral perspective, indicative of a lot of phenotypic variation. Once we put the machine learning model on top of it, we can make predictions of things like ploidy level. So here's a prediction of ploidy level across the landscape. Blue is diploid, red is triploid, and you can again see a pretty complex spatial mosaic here. This model has an accuracy of about 80, 85, or 90%, so it works pretty well. We can do it for sex as well, but our accuracy is closer to maybe 60 or 70%. So that's not as good of a model. But luckily for us, in the previous slide, you saw that sex was less important than ploidy level. So it doesn't necessarily kill us in the downstream exercises. So anyway, once you have all of these data sets and you've gone through all of this modeling, you can then put it into the integral projection model and then put it on the landscape. When you do that, you get a plot that looks like this over about 400 square kilometers of Western Colorado. The colors here show you lambda, and they're all red or gray, which in our color scheme means either stable or declining. So the big takeaway here is that there's not really many expanding populations of aspen. And in fact, there's actually quite a lot of declining populations. If you make a, and here's just the zoom, so you can see it a little bit better, just a lot of decline predicted. And if you make a histogram of all those lambda values with the value of 1 being shown here, you can again see nearly all of the values are less than 1, quite substantially in many cases. So I think the takeaway from this is that it's perhaps unsurprising that we're predicting decline, but I think this is actually very robust evidence demonstrating it from a population modeling perspective in a way that isolated measurements of mortality or growth wouldn't necessarily be able to tell you. And from this, we can also isolate the fact that some of this decline in some places is due to topography, but in other places it's due to genetics, and in some places it's due to the combination of these two things. So that's quite a useful conclusion. So the next thing you might wonder then is why are some of these genetic features implicated in death? And to get at that question, we need phenotype data. We need to go and figure out what's actually happening. So as a second prong of this research, we developed a number of different common gardens in which we assess the physiology of different Aspen genotypes. So since 2020, we went all across the continent and we collected about 300 different genotypes from across Western North America and also some parts of Eastern Canada, and then down in Mexico and North America as well. It took about 50 people to do all of this, but we brought the rootstocks back and we clonally propagated them into about 2,000 individual trees, and then phenotyped them, and then also tried to kill them in an experimental drought. To just give you an illustration of what this looks like, first you go out, and you typically have to go out in the winter because the hormone situation in the roots just doesn't allow for resprouting unless you get them in the winter time. Then you dig a big hole. It'd be hard if the ground is mostly frozen, and you cut out some roots, and then you bring them back and put them in trays and wait for them to sprout, and then excise the sprouts, and then eventually grow them up into larger trees onto which you can make measurements. So it's a very labor-intensive process. So if you do all the work, what sort of things do you find out? Well, I don't have time to show you everything, but I'll show you a few selected and more interesting findings from this exercise. And just to highlight for people who don't do this kind of work, the reason it's a common garden is that if they're all growing in the same conditions, then any variation in phenotype has to be due to genetic variation or due to you messing up the experiment, which is also a fair option. So let's see a couple of things. One of the first things we were curious about was photosynthetic rate. How fast could they possibly grow? So here we've made a contrast between diploids and triploids. And what we can see here is that Amax, a measure of photosynthetic rate, is higher in triploids, and in a mixed model, it's significantly so. So that's consistent with our triploids being faster growers. We also made some measurements of hydraulic conductance, and we also found that the triploids have higher conductance in their stems, indicative of a greater water conducting capacity. We don't have data shown for you here, but they're also a little bit more vulnerable to drought. That's another thing that comes out of the data, and typically trades off with hydraulic capacity in most physiological studies. And then we did this drought experiment. And we basically had control and drought treatments, and then we contrasted it for diploids and triploids. And what we can see in a mixed model here, and this is just a simple proxy of mortality. How fast do you lose all of your leaves? How fast do you ditch your canopy? We see that this happens faster in the triploids than the diploids. So that's also indicative of this cytotype dependent effect. We also find, I'm not showing it to you in one of these plots, that there's also faster resprouting in triploids. So they seem to die above ground, but then they also seem to come back faster afterwards. So it's like they're dead, but they're not really dead. They're just dead above the ground. So we measure a lot more traits. I'm not going to bore you with all of the plots one by one, but we can make a principal components analysis just to look at integrations. I'll show you two versions of this because depending on the traits you pick, you can put more or fewer points on the graph. So here's one plot where we tried to throw a lot of different strategy axis traits in. And we can see along our first principal component, there's this trade-off between do you really die versus do you resprout, for what I was talking about before. And then along our second axis, we seem to see a trade-off between do you have a lot of chemical defenses versus do you have a fast growth rate and do you have a lot of water conducting capacity. So this seems to be the main strategy trade-off here. And the diploids and the triploids, these different circles here, are loosely segregated along the first axis of this trade-off. If we then plot this slightly differently, climate versus resprouting ability, we see that climate actually seems to be statistically decoupled from this resprout versus not resprout axis, telling us that resprouting and not resprouting is something that you can do regardless of your climate of origin. That's sort of just a generic trade-off that some genotypes can do and some don't, but it's not like a Mexico only or a Canada only sort of process. So I think this is quite interesting. It gives us a little bit of insight into the different strategies that our different populations are having. It tells us that perhaps one of the reasons the triploids are dying so much is that they simply can't deal with these drought conditions nearly as well. However, they might not really be dead in a way that the integral projection model can find out. It might just be that they're temporarily dead and will perhaps later come back through resprouting. We don't have enough long-term time series data to know that yet, but it's something that's certainly worth finding out. So that work was all done on saplings between 0 and 3 years of age. And I think it's a fairly accurate description of reality for young trees. But we know that mature trees also exist and may experience different stressors than young ones. So the greenhouse at Berkeley is very expensive, and these trees weren't paying their rent very well. So we had to find somewhere else for them to go. And we were able to do a partnership with the Colorado State Forest Service out here to bring these trees here for a permanent planting. So we took all of our potted trees in Berkeley and put them in a really big truck and drove them out to Colorado. And then last year on Earth Day, we planted them, which turned out to be a snowstorm day. And it ended up being about 60 people's worth of effort, I think, to get all these trees in the ground, but we did. And they seem to mostly have survived the transport and the planting. And if you now go back, these are pictures from last summer. Most of these trees are alive and happily growing, and we're going to be phenotyping them and growing them for the next couple of years at least until the money runs out. So we don't really know what's going to happen to the bigger trees, but we hope to find out soon. So far, I've just shown you results for primarily cytotype, not really for sex so much, a little bit at the beginning about sex. One of the reasons we don't know more is that we haven't done the more detailed sequencing or PCR work it would take to either get the sex data or to look across the full genetic sequence rather than just at these bigger scale features. So the next big part of the project, which will be the second to last part of the talk, will be to talk a little bit more about all the allelic genetic variation, all the other stuff that's going on that might matter. So to get at this, we are producing reference genomes for the species. There is not really a good reference genome for Aspen yet. So we're producing it with the Department of Energy Joint Genome Institute. We're doing 2 reference genomes, and they're going to be haplotype resolved, which means that instead of just getting a single sequence for sort of a big mushy mix of both chromosomes in a diploid, we're going to be separating out what happens on each copy of each chromosome in that diploid, or on all 3 if it happens to be a triploid. And then the second thing we're doing is we're going to be doing something called whole genome resequencing, which is basically getting little short chunks of the genome, covering the whole genome for about 2,000 individuals across the species range. Our current sampling map is the black dots that you see here. So I think we have pretty good coverage of the overall range, minus some of these First Nations here up in central Canada, where it's very difficult to get roads or helicopters or anything else in. And this has been a really big partnership effort with a lot of different community partners, a lot of citizen scientists, herbarium records, and just many other things from which one can possibly get DNA. It's very humbling, I think, actually, to be able to put together a map like this. So where are the genomes coming from? The first genome we're doing is from the Mono Lake region in California. So this is in the Eastern Sierras. This is the tree. We just had my postdoc go visit it last weekend. And Yosemite National Park is sort of right over here. You go over the Sierras, you drop down the hill, and we're somewhere along the way down there. It turns out, and we know this now, that this is a diploid male that we're doing this sequence on. And that's useful because the males have the sex chromosome region. So we're gonna be able to see that. And then the second individual we're doing a genome on is Pando, which is in Utah, and we think is the largest Aspen clone in the world. Pando is a triploid and is a male. And it's quite big, but we had to pick one stem. And I felt like this one, so this is the one. And if you don't know, Pando is sort of in central Utah, and this location. And the Paiute tribe is here and or have some actively managed land very close to the Fish Lake National Forest where the tree currently is. So we're still getting a lot of this data back. We have about 300 of the 2,000 individuals for the sequencing work done, and one of the 2 genomes done. I can just show you a couple of very quick results from what we have so far as a very preliminary story. The first thing is what's going on in the genome. How does the Aspen genome look to other related Populus species? So this is for our California one, the diploid, and we actually got a very surprising and unexpected result for this. So you can do something called a scintillating plot, which basically shows you how do the chromosomes in this species, all the genes, how does everything match up to other related species or to one haplotype relative to other. Our genome is these 2 lines here. So this is basically one set of chromosomes for our California tree, and this is its other set of chromosomes. Up here is Populus trichocarpal, a very well-known Populus. Down here is a cross between Tremula and Alba, and then here's another haplotype for Tremula and Alba. These big swoopy sort of, I don't know, pillows or ribbons or whatever you would like to call them, show you the matching between the different chromosomes or the different species. The most obvious thing that stands out here is that in our California individual, chromosome 1, which is the biggest chromosome, is not actually the same in both of them. Chromosome 1 is actually on one of the haplotypes sort of a smushed up mix of what chromosome 1 and 10 would be in every other related populace in the other half. So this is a very big structural variant that we're seeing here. And the fact that we got it on our first try without really looking for it suggests to me that perhaps these large structural variants might be very common in other locations as well, and potentially could have very big implications for gene regulation and expression just due to the physical position where these genes happen to be. So this was very interesting. We don't know anything about what Panda looks like relative to this yet, but we hopefully will in another month or two, and I hope to share that with anybody who's interested later on. So the next thing we want to do is just all the sorts of standard population genetics and genomic analyses you can do with variation at different sites. And for this, you typically have to follow some sort of big pipeline where you have your short read sequence data, you have a reference genome, you do a bunch of things to get them to match up to each other, make sure you're happy, and then you can do something called calling variants to figure out where all of the different single nucleotide polymorphisms are. With this data, we can also, we think, determine ploidy level based on allele frequency ratios and based on the presence or absence of the sex determining region on chromosome 19. We can also call them as male or females. This is what my postdoc is doing right now, and we haven't done it yet, but we're in progress. And it's computationally quite intensive, as you might imagine. And we have a couple thousand genomes to do this for, so it's going to take some time. But we'll have this soon, and I think things are going to get exciting. But for the 200 or so that we have been able to do this for to date, we're able to look a little bit just about some broad kinds of genetic variation across landscapes. So you can do a principal components analysis of the genome in the same way you could for any other data set. And here's a PC1 and a PC2 plot. PC1 has a very surprising pattern, which is that California and the Cascades are just totally different from everybody else, which is not something we expected. My hypothesis personally is that this is that big structural variant that you saw just being locally present in California, but not present anywhere else. Don't know if that's true. We'll find out soon. And then PC2 seems to be really a latitude dependent sort of thing, where the Southwest, and we don't have Mexico yet, we hope to soon, seems to be very different than these more northern locations. So what does this really mean for functioning and phenotype? Don't know, but we hopefully will soon. So to wrap up the second to last part of the talk, what can we learn from all of that? Well, I think one of the clear things that we can immediately see is that if you're a triploid, you're very likely to have this fast and risky and resprout strategy. You grow fast, you use resources quickly, you die easily, and potentially you resprout better than a diploid. And everybody has a hard time when it's hot and it's dry. Females also seem to be at greater risk than males in many situations, which is really interesting sexual reproduction implications. And then lastly, just based on what we've got so far, there seems to be a lot of structural and then also just variation in these genomes across the range. Once we have more of this, we're going to be able to play a lot of games where we can do genome-wide association studies to see which genomic regions are associated with certain traits that we've measured, and then these genome environment association analyses to figure out how adapted or maladapted these populations are. I can't show you yet, but probably in about a year, I think I'll be able to. So now in the last part of the talk, I want to just highlight some more thoughts than results about conserving genetic diversity in Aspen. We know from these big species distribution models that many populations, especially those on the trailing or the rear edge, are likely to be lost in the future, at least are at risk of being lost. And we also know from a lot of evolutionary biology and empirical studies that in many species, the rear edge often includes a lot of important adaptive genetic variation, which might be especially important to preserve for breeding efforts or assisted migration efforts. So what's happening on the rear edge? We don't really know in general, but in my group, we're starting to at least begin to focus our sampling on some of these locations. And we don't have cash yet to try and specifically collect root tissue from these places and preserve it. But we're hoping to do that in the future. But I'd like to at least illustrate for you a little bit of just what some of these trailing edge populations look like. One of the first I'll show you is in the Yala Valley Mountains, which is a word which translates to snow mountain in English. Some of these are very snow-covered mountains in the southern Klamath Range. This is a location which is the very southernmost extension of the Klamath Mountains in Northern California before you drop out and get to much lower elevation locations. So it's sort of like a very isolated location. If you look at this photo here, it looks mostly just like a burned conifer forest, and indeed it was burned a couple of years ago very severely. I went there last summer with a botanist from the Forest Service, and we went looking for aspens to see if there were any that survived. And you don't really, immediately see anything, but it turns out this is actually an aspen tree up on the slope, and it's growing out of a stream which is snowmelt fed from these upper locations. So it's just hanging on with a little bit of water from the runoff on this slope. If you walk down the slope, which is a great idea going downhill and a much worse idea coming back afterwards, you do see perhaps maybe 200 or 300 more stems. So it seems to be actually a pretty decent population here. It seems to be doing okay. Might be doing just fine, but it sure is lonely. A second one, San Gorgonio. I showed you a picture before. Here's another photo. Some of you may know this is not the fires in January, but the previous summer there were some big fires in this location that passed over this location. We don't know how these aspens are doing. I hope they made it. They probably did. I don't think it was a very intense fire there. But we've tried to at least do some DNA sequencing from this a couple of years ago to figure out what they look like. It's a very isolated stand up in these mountains by Los Angeles. There's also some aspen in the lower Midwest, believe it or not. And I was able to find this interesting herbarium record from the 1930s on a private landowner's parcel, which you can see in this satellite image here from the present day. I was able to do a bunch of phone calling and meeting people in this community. And the best I was able to hear from a guy who works for the Forest Service, David Massengale, is that this tree died in the late 1930s, and that was it. And there are no longer any aspen in Missouri. If you look at some of the Forest Service range maps, they still have a polygon there, but that's entirely due to this bit of evidence, and it's dead, no longer there. And the herbarium sample is a very small and degraded bit of stem, and that's it. So we don't know anything about it. And then it's in southern North Carolina too, believe it or not, along the French Broad River. This is what it looks like. This is not the natural population, but actually a shoot, which is growing in the backyard of a gentleman named Ron Lance, who is a guy who likes plants a lot. And in the late 1970s, he observed this along a river corridor and thought, this looks really weird. I don't know why this is here. And he had the foresight to take a cutting and plant it in his yard. The natural population then died some years afterwards, and this is the last remnant of it. And we are trying to sequence this one. Southern Arizona, in the Sonoran Desert, sort of on the Sonoran transitions. Some of you may have been to the Galiura Mountains. This is what they look like. It's low elevation, like 4,000 feet. It's really, really hot. And if you climb Bassett Peak, which is this mountain, you'll notice about a third of the way up, there's this little aspen stand in a little valley down there. It's totally isolated from any other aspens by a good 50 or 60 miles at least. And the whole population, based on my count, is about 10 adult trees and about 3 saplings. And that's it. You can see one of the adult stems here. And I think the reason they're here is that there's this fracture in the rock here. And I think there's some subsurface water flow, which is subsidizing this population. They don't look like they're doing so hot. I don't know if they're going to be there in another couple decades. And then what about down in Mexico? So we think this is the southernmost aspen in the world, down in Jalisco. This is a photo taken by our local collaborator down there. You can see it kind of looks cottonwood-like to me, at least. It doesn't really look very aspen, but it is an aspen. It's growing in a stream bed here. It's growing right next to a power transmission line, and there's a road right over here. So who knows what's going to happen in the long run, but it is there for the time being. And then also, here's another photo. This is Querétaro, different state. And this is primarily a conifer forest, but there's a couple little isolated stems there. This is most of the population. So there's a couple little aspens hanging on. They seem to be doing okay, but there's not many. And this is, a lot of the Mexican aspen diversity for the time being. So one thing we are trying to do with an eye to the future is we're specifically trying to propagate Mexican genotypes in the US to sort of create a living library of genetic diversity and have at least some representation that stands in other locations. We explored doing it in Mexico, and it was a little bit complicated to think through those logistics, but we were able to work with a collaborator at a university in Guadalajara, and he was able to send his team out. These are 2 of his botanists. They're digging some root samples for us here along this aspen stand, and we were able to get them exported under legal terms back to the US, and my student here is taking those roots, and we're propagating them up into adult trees to hopefully have on hand for the future. So I think this maybe leaves us with a couple of questions that at least I'm thinking about, and I'd be curious about your opinion about. are there any prospects for improving regeneration in these ranged populations? For example, that Galliura mountains population, it's got 3 saplings and that's it. Can we make that 5? Can we make that 20? Can we get that one back? Can we, in ways more substantial than my small bit of cash can allow, can we invest resources to get some of these ranged populations? So, perhaps a zoo or a library sort of situation, or can we do things in situ? And if we are thinking about these genetics, are we really thinking towards an era of assisted migration or genomics assisted breeding programs? Is this even a good idea? And in any of these cases, if they aren't, can we also find ways to do more restoration based on seedlings? Can we get sexual reproduction and generate some genetic diversity and produce that at scales in a way that might actually be useful for restoration efforts? In California, at least, there's a lot of potential buyers in the nonprofit land, but no one's selling because no one knows really how to do it yet. We don't either, but I think it's an interesting question to consider. So to end the talk, I told you, this is maybe a genetic perspective on the future of Aspen. I don't think it's an answer about the future of Aspen, but it's at least a couple of ideas and lenses to think about the problem and some, I think, open questions to consider as we get to a reckoning for the species. And perhaps not all, but many of these questions are relevant, I think, for work on other important forest trees here as well. So I hope it's a thought-provoking look at some of this for you. I'm going to end the talk there. We have a lot of funders to thank. A lot of different people helped us with permits, and numerous people, more than I can list here, have helped with the fieldwork, the analyses, the logistics, everything else. So thank you, and I'll take questions.
Do you know for having sex in tremuloides is kind of trippy to think about? Is there a higher ratio of male or in triploids that you know?
So you're asking about offspring of triploids?
Maybe not even offspring, just on the landscape. Or do triploids tend to be either male or female?
Yes, that's a great question. So what's the relative frequency of triploid of males and females given that you're triploid or diploid? We've done the cross tabulations in our data sets. They seem to be independent axes. It doesn't necessarily seem to be more that if you're a triploid, you're more likely to be male or vice versa. And that's interesting because thinking about the way the sex determination region works, it could have gone the other direction, but it seems to be independent so far. However, with our much broader, so that's based on a couple hundred samples from Colorado. Mm-hmm. With our new data set, we'll have 2,000 individuals from across the species range, and then I'll have a better answer for you, but I don't know what that answer is yet.
Thank you. That was a really interesting talk that represented quite an impressive amount of fieldwork. I'm curious, based on Colorado plots, if you were able to incorporate things like disturbance history, which might relate to resprouting or intra-specific competition, machine models.
Yeah, that's a great question. So can we incorporate any sort of land management factors or disturbance history? In the deep sense, we don't know the disturbance history of the land well. We know that the area was open to silver mining in the late 1870s and was very heavily used in the 1880s for that purpose. We know there was a lot of deforestation for powering the mills and everything else, but we don't have maps, unfortunately. So from a quantitative perspective, we don't really know where people were using the land. More recently, we do know where grazing is occurring based on Forest Service regulations, and we also know a little bit about where recreation is occurring. We haven't explicitly put those predictors into our models so far, but we have in previous demographic modeling work assessed overall herbivore presence based on a combination of scat and trampling and nibbling that we observe in the field plots. And that in previous models seems to be a very good predictor of recruitment, unsurprisingly, where the higher that is, the lower the recruitment is. I would think that would then, in the models you saw now, have a big impact on the lambda estimates we produce. One of the reasons we haven't put it in the model is that we don't think we can upscale those measurements to the landscape very effectively. So we're just keeping in predictors that we think are upscalable.
So I thought the south-facing slope part was really interesting, but I was wondering, is that happening at the less frequent because of less genetic diversity, or more because of the climate stressor, or maybe both?
So the question is, are we seeing the dieback on the south-facing slopes because they're more likely to be triploids, or because of the climate stress, or both?
Yeah,
So what we do know is that in the common garden studies, we know that the triploids suffer more. So that's a very, very clear argument that there is this drought by cytotype interaction. But we do know also from the field data that everybody just dies when it's hot and dry. So I think the answer is really both in this case. But you also asked about overall genetic diversity, and we haven't really mapped that up very effectively yet. That's something that we're hoping to do better in the future, and maybe tractable to remote sensing, but that would be a different question. Yes, and the blue shirt. I think what I was referring to, let me go back. I think I was referring just to the integral projection models. Is that your question?
Yeah.
So for people who have done like a matrix population model before, this is sort of a fancier version of a matrix population model. The idea is that a population is built of individuals of different states. And those could be like different ages or different sizes. And as time goes on, individuals progress from one size or one age into the next size of age. So what we can do is we can build regression models which predict the probabilities of those transitions based on size-dependent survival, size-dependent recruitment, size-dependent growth, age-dependent versions of all of those, and then potentially also on other covariates like the environment or the genetics. So what we've actually done under the hood here is we've fit a lot of different linear mixed models based on, which are regression models based on all of those different factors. And then we've combined them through the known mathematics of integral projection models to produce something called a kernel. And then once we have this kernel, we can then get the eigenvalues of the kernel. It's just like a very big matrix. And when we have the eigenvalues, that tells us the lambda values. So that's really what's happening under the hood here. It's actually a fairly standard approach, but it's quite a pain to implement in practice.
Kind of like looking forward from John's question about south basin areas. I found it interesting that the triploids seem to use more water, they seem to grow more, and in my tree or forest ecology sense, usually if you have more growth, those trees, or if you're growing more, you have more of a capacity to expand, but that doesn't seem the lambda values for facing areas where you have you're more resources to be able to grow didn't really result in any population expansion. I was just curious if you had any theories as to why that might be.
Yeah, that's a great question, and it actually follows perfectly to the previous question. So it's true that we have evidence that triploids grow better, but growth is only one component of fitness. Other components have to do with mortality and with reproduction. So it turns out that while they grow better, they also die more easily, and they recruit less easily. And it turns out when you do all the math, the mortality and the recruitment sort of swamp out the growth. So even though they're growing better, the other demographic rates sort of kill them in the end.
Despite having more water and less likely,
Yep.
or lower likelihood of drought, that still got swamped out.
Yeah. And it could be that the reason that they do less well at recruitment might have nothing to do with water. It might just be that they invest less in defense, so they're tastier, so they get eaten by a deer, and that's the end of it. We can't distinguish that in this model, but the implication is that something makes them less good at reproducing.
Yeah, just to like follow up. I wonder if you think like that pattern might be different outside of South as well. Like in areas that are less water limited, potentially like those
Yeah, that's a super reasonable question. I wish I knew the answer to that. In my group, it has taken all of our effort to get to this result. But if anybody would like to do a similar study somewhere else, I think the comparative ecology of that would actually be really interesting. Unfortunately, a lot of the sort of big standard forest inventory data sets that people at, for example, the Forest Service collect are not necessarily suitable for this because they don't always, and not always, but often don't include some of the key recruitment factors or the direct observations of mortality to be able to pull this off. But in some cases they might, and then it's just an issue of getting someone to fit the model and see what happens. They also would need to have genetic data, which is the expensive part. I think we spent probably $30,000 or $50,000 trying the genetics for that study, and someone would have to find the cash to do it in the other places as well.
Do you have plans to sequence the ultimate relic population, which is hybrid between tremuloides and dentata in the Niobrara drainage.
So are we planning to do any hybrids, for example, in the Midwest? We think we have leaf tissue from those from a collaborator. We haven't thrown it into sequencing yet because our expenses are basically getting paid by DOE and they don't want any hybrids. They're not interested. So I think we might be able to do it if we did it on the down low, but we're not supposed to.
I know a guy that
If you know a guy, we can help provide the tissue to do that down hand.
The increase in K max, like higher hydraulic conductance in the triploids, do you know if that's due to larger vessel diameter or just?
We sort of know. So we have measurements from field populations of that, and they do look like they have larger vessel diameters in the field, but we haven't done it in the common garden. So we don't know in that sense. But if you at least believe that there isn't a big environmental or ontogenetic effect, then it's probably true.
So yeah, they just do a very good job.
Mhm. Yeah, that was an undergrad project and we only had enough effort to get that far in the field. Okay, well, thank you everybody for coming. I appreciate your attention.